In the previous section of this course, we began to discuss the nervous system, specifically the neuron and how the neuron can send a message, an electrical message, from one side of the body or one side of an organ to another side. Now we're going to kind of zoom out and start talking about the nervous system. So talking about nervous tissue and what other types of cells are involved, what other types of structures there are. Specifically, we'll be looking at part of the central nervous system, the spinal cord, and how the spinal cord can also send messages back and forth to the brain, and it is taking in sensory information from the periphery of the body and sending out motor commands to those peripheral organs. This section covers quite a few different chapters, and you can see those chapters on the screen ahead of you. Parts of chapter 7, 11, 12, and 13, and hopefully we can bring all of these together to give you a pretty good indication of what's going on with the spinal cord, where it is, how the body protects the spinal cord, and all the very important functions that it does. Okay, so to pick up from where we left off at the last section of this class, this here is a neuron. Right, as we know, the neuron has dendrites, right? So these are dendrites there, the receptive ends of the neuron. Ultimately, they can be depolarized or hyperpolarized. All of these postsynaptic potentials can be summed up here in the cell body or the soma, and if the depolarization is enough to exceed threshold, an action potential will be generated in the axon hillock and ultimately travel all the way down the axon to release neurotransmitters into the synapse to control a target cell. We briefly discussed, again in the last section of the class, that there are different types of neurons. For the most part, we drew this type of neuron right here. Okay, this is a unipolar neuron, and it is a unipolar neuron because there is one or uni extension off of the cell body. Okay, so we can see the dendrites and one single axon, and the neuron cell body is actually kind of an appendage off of that single axon. Okay, so that would be right here. So unipolar, these are mostly sensory neurons in the peripheral nervous system. For the most part, they are carrying a single message, from many, many different places. Okay. The other type of neuron that we have looked at relatively extensively are multipolar neurons. Our model in the lab is a multipolar neuron. Most of the diagrams in your textbook and on animations online, they are all showing you multipolar neurons. And so a multipolar neuron has multiple appendages or multiple branches off of this single cell body here. And so again, all of these dendrites are individual extensions, and then axon hillock and one single branch, the axon. Okay. These are by far the most common cells within the body, within the nervous system. They are motor neurons, right? So we could be sending lots of different messages to this single cell, right? Lots of different decisions made by the central nervous system, but ultimately there's going to be an action potential or not. That is, there's going to be a muscle contraction or not. Okay, so lots and lots of integration here. Furthermore, multipolar neurons are found to be interneurons, and we'll talk about interneurons and how they are a functional class of neurons in a couple minutes. But in general, these interneurons are essentially integrating information from the sensory side of our nervous system, right? So what is the temperature, what is this thought, what is that thought, what are the environmental conditions, and ultimately making a single motor decision. Okay, so those are interneurons. They are found exclusively within the central nervous system. So again, multipolar neurons are by far the most abundant of all of them. Okay, about 99% of neurons in our body are multipolar neurons. Okay, so multipolar are the most abundant. The least abundant are bipolar neurons. And bi indicates two, and so off of the cell body there are 1 and 2 branches. These are the most rare because they are only really found within the special senses. Okay, so your photoreceptors within your retina of your eye, they communicate with bipolar neurons. Bipolar neurons are found within your ear, just very specific special senses. Okay. So unipolar, bipolar, and multipolar neurons are the three options within our nervous system. These are considered to be structural classifications. Okay. For all neurons, regardless of whether it is uni, bi, or multipolar, all send information in one single direction, right? So here receiving information and here sending information. There is no bidirectional flow of information. Okay. This is an image representing the entire nervous system. Okay, we're going to see this again and again throughout the rest of the semester, but really this gives you the big picture view of where information is coming from, where it is going, and what divisions of the nervous system are being utilized. Okay, so let's start down here at number 1. Okay, what we'll be focusing on a lot is sensory information. And what we can see here is that there are a variety of different types of sensory receptors. Somatic sensory receptors would be found in places like the skin, right? Essentially detecting touch, right? Pressure, pain, temperature sensation. So all of these different types of receptors present within the skin and maybe some other visceral organs to figure out what is going on in those peripheral locations. There are also visceral sensory receptors, right? Monitoring things like blood pressure, pH, gas concentrations, so those types of things. And then the special sensory receptors. Yeah, the special senses we'll talk about at the very end of this semester. They essentially utilize highly modified and highly localized receptors. Okay, so many different types of sensory receptors, all of these detecting something very specific because of the specific structures within the dendrites, okay, ultimately leading to depolarization, exceeding threshold, and the generation of an action potential. That action potential is going to travel along axons bundled into peripheral nerves. Okay, so all of these nerves are in the periphery, right, detecting information not within the central nervous system, but from the rest of the body. Okay, and ultimately that information about how you stub your toe is going to travel along axons within peripheral nerves, ultimately to communicate with the central nervous system. Okay, the central nervous system is where all of this information is then processed. Okay, so not just detecting things, but actually deciding how to respond, right? You're kind of making sense of this information, right? Yes, we got an action potential all the way from the toe, but what does that mean? Okay, so the central nervous system is going to compare this new information with memories of previous times you have, for example, stubbed your toe. Okay. The central nervous system consists only of the spinal cord, which of course is the function or the focus of today's lecture, as well as the brain, which we'll talk a lot more about later this term. Okay, so the central nervous system, whether we're talking about the brain and or the spinal cord, processes information and ultimately makes some kind of a decision on how the body will respond to this information. And so the motor division of the peripheral nervous system is going to carry out these motor commands as determined by the central nervous system, right? You stub your toe, okay, how do we respond to that? Do we pull our foot away? Do we grab our foot in pain? What exactly are we going to do? Okay, there are different types of motor commands and ultimately different destinations. What we'll focus on most this semester is the motor commands carried along carried within the somatic nervous system. And the somatic nervous system is controlling your skeletal muscles, so your conscious movements. Do you grab your toe? Do you not grab your toe? Compare this with the autonomic nervous system. Okay, we'll learn just a little bit about this by the end of the semester, but for the most part, AMP2 covers the autonomic nervous system in extensive detail because the autonomic nervous system regulates your visceral organs. Okay, so your smooth muscle, for example, so what are your intestines doing? What is your stomach doing? Your cardiac muscle, right? Your heart, and glands and adipose tissue. So what are we secreting into our blood? What are we secreting into ducts, onto the skin, etc.? Okay, so keep in mind that sensory information is traveling via action potentials within neurons to the central nervous system. The central nervous system processes this information, produces some kind of a motor response or an executive decision. Then that motor decision is carried again into the peripheral nervous system via the somatic nervous system to our skeletal muscles. Just like we had structural classes of neurons, right? So we have unipolar, bipolar, multipolar neurons. We also have functional classes of neurons, and essentially the functional classes just are determined by where in that pathway these neurons are located. So for example, sensory neurons, we have about 10 million of them in our body, so of course these are very important for us. The more sensory neurons that we have, the more our body can kind of know how to respond to the environmental stimuli around us. Okay, and so in this example over here, we have the brain and the spinal cord. That is our central nervous system. Sensory neurons are going to be detecting something in our body, in this case blood pressure, by detecting stretch in the aorta. Action potentials travel along sensory neurons to the central nervous system for processing. Okay, again, in the skin, we have this sensory neuron ultimately detecting some kind of a stimulus within the skin, sending an action potential along its axon, again, to inform the central nervous system of what exactly is occurring within the skin. Okay, so those are sensory neurons. Interneurons, as I said before, are within the central nervous system. These are the ones that are actually processing the information. They are actually comparing, this data with previously stored memories, with pre-programmed responses, maybe even allowing us to consciously think about how to respond, right, to whatever sensory stimuli just came in. And so these interneurons are by far the most abundant. Right, we can see up here about 20 billion of them in the body. Meaning that this processing, this integration, is critically important to the human existence. Okay. Finally, these interneurons are going to make this executive decision and send out the information along motor neurons. Okay, and there are far fewer motor neurons because, again, you either move your foot or you don't. Okay, so here we can see different types of motor information going to the heart, and again, different type of motor information going to the skeletal muscles. Right, this is part of the autonomic nervous system. This is the somatic division of the peripheral nervous system. Okay. Not all information processing by these interneurons is going to be generated exclusively by the brain. Remember that the spinal cord is also part of the central nervous system, and so sometimes for certain responses, we actually have pre-programmed responses that are mediated by the spinal cord alone. And so one example that we're going to look at in more detail later is this right here. This is a cross-section through the spinal cord and, of course, the two legs. Note that the foot is stepping on something painful, right? Broken glass, this hurts. Particular receptors, so sensory neurons, are going to detect this painful stimulus, and action potential travels all the way up this neuron, okay, ultimately entering into the spinal cord. Now, instead of immediately sending this information up to the brain so the brain can say, should I move my foot or should I keep stepping on this very painful broken glass? Instead, this sensory neuron immediately synapses, which we can see right here, that's what this is indicating, immediately synapses with these green neurons, these interneurons. Okay, and these interneurons are pre-programmed to produce a particular response in or whenever this painful experience is experienced. So what we can see is an immediate synapsing with motor neurons. And so these motor neurons are going to tell the muscles of the other leg, you need to contract, right? You need to prepare for all of the weight of the body to be on this single leg, because on the other leg, we are going to contract our muscles in such a way that we pull our foot away from that painful stimulus, lightning fast, now just standing on this particular leg. Okay, this is called a reflex, right? Something stressful, something painful happens, and immediately you respond in order to save your skin, save your tissues. Okay, so this is just one example of sensory going to, interneurons processing within the spinal cord and or the brain, and finally motor response immediately going out. Okay, one more term or set of terms to mention for you guys. We can say sensory neurons, indicating that they are going to the brain, like carrying sensory information, but we can also use the term afferent, which I will hereafter say afferent, because afferent is exactly opposite from efferent. Okay, afferent means going to the central nervous system. Efferent means going away, like exiting. Afferent and efferent sound very similar to each other, so I will emphasize afferent versus efferent. Okay, so you will see both of those terms, sensory and motor, and afferent, efferent, used interchangeably. Okay. So motor neurons can synapse with a variety of different tissues to produce whatever motor response was deemed appropriate by the central nervous system. First of all, what we've been talking about is a neuron synapsing with another neuron. So that is one way that we can produce a motor response. Secondly, we can send an action potential along this efferent neuron to trigger some kind of response in a skeletal muscle. Right, that will be very important in this section of the class. Finally, motor responses can be produced by neurons, motor neurons, efferent neurons, synapsing with glands. Okay, so this point we've talked a lot about neurons. We know that they are taking information to the central nervous system, within the central nervous system, and away from the central nervous system. So neurons themselves are the functional units of the nervous system. However, they are by far not the only types of cell that are important for the function of this system. Okay, in fact, the nervous system is comprised of nervous tissue. Right? Just like we know about any other tissue, there are specialized cells that function together for some kind of a greater purpose. And so nervous tissue that is, contains neurons, of course these functional units, but also there are many supporting cells within nervous tissue. These cells are collectively called neuroglia. Glia is a term that means glue, and so previously anatomists and physiologists thought that these extra cells were just kind of gluing the neurons together. Of course, at this point, we know a lot more than that. It's not just glue, it's not just holding the structures of the central nervous system together, but in fact, a lot more specific functions as well. Okay. We actually already know about one of these two of these neuroglia. In the peripheral nervous system, as we know, the myelin sheaths are composed of Schwann cells. And again, as we know, a single Schwann cell, right, filled with this fatty substance that serves as a great insulator. These cells wrap around the axons of peripheral neurons again and again, so kind of like paper towels or toilet paper around the cardboard tube, the axon being the cardboard tube, and the cells themselves, or the Schwann cells themselves, being the paper towel. Okay, so ultimately, many, many layers of this insulating substance. Okay, Schwann cells each myelinate a single axon, not multiple axons, and they are separated, of course, by these nodes of Ranvier. And of course, these nodes of Ranvier are where the action potentials are regenerated, producing saltatory conduction. Okay, so we've already seen Schwann cells. However, there are more functions to Schwann cells than we previously discussed. Okay, yes, they serve as great insulators. Yes, they increase the speed of action potentials, but also they protect the axons. Okay, so they serve as kind of a buffer, keeping these very delicate structures away from the surrounding interstitial fluid, so the fluid surrounding all of our cells. Because of this protective function, some axons that are non-myelinated, right? So they do not have this saltatory conduction, but they are associated with Schwann cells. Okay, so as it says here, Schwann cells can be found around unmyelinated neurons. As we can see in this diagram over here, here is one neuron cell body and a series of other axons from other neurons. The Schwann cells are essentially grouping these extra cells together, or many axons at the same time, and essentially protecting them from the surrounding interstitial fluid. The reason why these axons are not considered to be myelinated is because there are no nodes of Ranvier. Right? They are not individually wrapped and therefore individually insulated. Instead, they are just kind of grouped together by these Schwann cells simply for protection, for stabilization. Okay. Peripheral neurons, right? So these sensory and motor neurons within our extremities, they are also associated with a second type of neuroglia called satellite cells. And we can see the satellite cells up here around the neuron cell body. These are surely the most poorly understood of all of the neuroglia. However, at this point, we do appreciate that they are regulating the environment around the neuron cell body. For example, they are regulating ion concentrations so that, for example, if potassium gets too high in the interstitial fluid, they can take in some extra potassium ions and therefore help to maintain that resting membrane potential in the neuron. Okay. There are similar cells within the central nervous system called astrocytes that are much better understood. Okay. The central nervous system. There are many more types of glial cells. Of course, we need lots of different types of cells to make up the brain and the spinal cord, and of course a lot of protection within it. The first type of glial cell within the central nervous system are the ependymal cells. The ependymal cells are essentially highly modified epithelial cells. They have cilia on them, and essentially they create a current, they create a flow of central or cerebrospinal fluid. Okay, so if we take a look at the spinal cord, as we're about to learn in this lecture, the spinal cord is housed within the spine itself. There is a series of holes through our vertebrae that connect to form a nice canal, a nice vertebral canal, in which the spinal cord is located. If we take a section through that spinal cord and spine, what we can see is here are the bodies of the vertebrae pointing towards our belly. Here is the little process, the little extension off of a vertebra that we can feel through the skin of our back. And between them is the spinal cord. Okay, there is some space around the spinal cord as well as within the spinal cord that is filled with something called cerebrospinal fluid. And specifically inside the spinal cord, again, here's the spinal cord, a section through it. There is a canal called the central canal. Okay, and this is lined by ependymal cells. Okay, the ependymal cells essentially create a current. Here we can see their little cilia, and they're going to wash the CSF up and down that central canal, essentially helping to deliver nutrients, creating buoyancy, pulling wastes out of the nervous tissue. So again, these ependymal cells assist in producing, circulating, as well as monitoring the cerebrospinal fluid. Microglia. These cells are, as it says, embryologically related to monocytes and macrophages. So essentially, these little furry-looking cells here are constantly on patrol. They're kind of wiggling in between all of these different cells. They use these little branches to phagocytose, so literally to Pac-Man, to eat anything that doesn't quite belong. This could be a bacteria that somehow wound up in the central nervous system. It could be fragments of neurons or other types of cells, proteins, anything that doesn't quite belong. These microglia essentially go around and they vacuum up this extra stuff in the CNS. Okay. Not a glial cell, but an important type of tissue within the central nervous system is called gray matter. And I bring it up now because this image that we've been walking through is the gray matter of the central nervous system. Essentially, this is the region of the CNS that is containing mostly neuron cell bodies, right? And therefore the dendrites as well as these synapses with other types of neurons. So all of the actual synapses, all of the true integration is occurring within this gray matter. right? It's called gray because these areas, right, or these parts of the cell are not myelinated. Here we can see one myelin sheath, but the region we're talking about does not have an excess of myelin sheaths. Right? Again, for cell-to-cell communication, so where really the magic is actually happening. In the central, or in the spinal cord, we can see that there is almost a butterfly shape of gray matter surrounding the central canal. Okay, we'll see that in more detail later. Okay, now we have, zoomed out. Right, so this is the same image that we've been walking through. Here are the microglia, here is a neuron cell body, dendrites, and these are synapses with other types of cells. So all of this is gray matter, okay, and of course the central canal lined with ependymal cells over here. Now we have zoomed out, so now we can see the white matter as well. Okay, the white matter contains these myelin sheaths, so myelinated axons, as we can see down here, white matter, gray matter. Okay. The third type of glial cell, specifically within the central nervous system, are called astrocytes, astro indicating star-like. So if we look at these cells right here, we can see that there are so many little appendages off of these cells, they look a little bit starry. Okay. The astrocytes are kind of a jack of all trade. They do so many different things in the central nervous system. First of all, maintaining the blood-brain barrier. Okay. You've probably heard of the blood-brain barrier before. Essentially, this is a highly selective barrier, letting very few substances cross from the capillary. Right, so this is a blood vessel right here, across the endothelium of the capillary, across this astrocyte barrier here, and finally into the central nervous system. Because your CNS controls pretty much every function in your body, it is critically important to protect the CNS from anything foreign, right? From any kind of shifts in ion concentrations or anything that doesn't quite belong. So these astrocytes are what are responsible for making the blood-brain barrier. Okay, astrocytes also kind of provide that glue, that glia. They provide structural support. As we can see, these astrocytes have little appendages stabilizing axons, right, multiple axons at the same time, also kind of holding these blood vessels in place. Okay, so lots of structural support is provided by the astrocytes. Just like the satellite cells in the periphery, these are going to regulate the extracellular environment, or this interstitial fluid. They regulate ion concentrations, nutrients. They kind of help to get rid of any waste produced by these neurons. Okay, so lots of maintenance produced by these astrocytes. Also, the astrocytes are going to help absorb and recycle neurotransmitters. Okay, so just like we talked about with the acetylcholine, we don't want these neurotransmitters that are released from synaptic vesicles to just kind of hang out in the synapses. The longer they're in the synapses, the longer they are triggering action potentials or inhibiting them in the postsynaptic cell. And so we want messages to be temporally linked to the stimulus itself or the decision itself. Okay, so astrocytes are also involved in that. Finally, astrocytes help to form scar tissue after a central nervous system injury. Okay, so what we can see down here, a little bit more detail than you need, but what I want to point out here is that when a neuron is damaged, right, so when you get a concussion and your axons are stretched or they're torn, it is your astrocytes in the central nervous system that can essentially use these little feet, right, so these little feet-like appendages here, to overlap and pretty much put a band-aid over your axon as it is regenerating, as it is growing back, which is possible. It just takes a really long time. Okay, so astrocytes are helping to heal your CNS. It's actually the the Schwann cells that help you to heal damaged nerves in your periphery. Final glial cell in the central nervous system are the oligodendrocytes. We've talked about these before. These are the myelinating cells within the CNS. Unlike the Schwann cells, oligodendrocytes can insulate multiple axons at the same time. Okay, so we look down here. Here is one oligodendrocyte cell body, and we can see multiple little appendages wrapping around multiple axons simultaneously. Again, different from Schwann cells, which can myelinate only one axon at a time. Okay. The abundance of myelin, or this lipid insulating substance, is what gives white matter its white appearance. Okay. And you can see this white versus gray matter with the naked eye. Summary of all of the different cell types that we're talking about in this class present within the nervous system. All of these cells up here are only present in the central nervous system. We have our astrocytes doing all sorts of different maintenance. Microglial cells, essentially your immune representation within your central nervous system, kind of vacuuming up whatever doesn't belong. Ependymal cells producing, monitoring, circulating cerebrospinal fluid. And finally, the oligodendrocytes, the myelin sheaths of the central nervous system. In the peripheral nervous system, we only have two cells. We have the satellite cells kind of supporting the neuron cell bodies, and then we have the Schwann cells producing those myelin sheaths. So that is the tissue that makes up, or the cells that make up the tissue within the nervous system. There are additional levels of organization, of course. This tissue is organized in a very predictable manner to make up peripheral nerves. Okay, so as you can see up here, the peripheral nervous system contains nerves. They always are organized with the same general pattern. Okay, and that pattern is shown for us down here. What we can see is an individual myelinated axon, right? So that is where action potentials are actually traveling along the level at which we have been discussing the nervous system thus far. Okay, however, individual neurons are not in isolation. Okay, they are organized and bundled into these nerves. And so each individual neuron is surrounded and protected by a connective tissue sheath called an endoneurium, and we actually saw this on the model in the lab. Okay, endo, meaning on the inside, all the way on the inside, neur, N-E-U-R, indicating something to do with the nervous system, in this case, around a neuron. Okay. All of these individual cells, right, are bundled together, right, each with their own individual endoneurium, and they are wrapped in a sheath called a perineurium. Okay, so peri for around the perimeter of this bundle of neurons. Okay, the bundle itself is called a fascicle. Okay, furthermore, multiple fascicles are bundled together and wrapped around by this connective tissue sheath called an epineurium. Epi meaning on top of or upon. So this is all the way on the top of this bundle of neurons. Okay, you can kind of think about this organization, this nested structure, like eggs, right? So an individual egg, the nutritious part is all the way on the inside. It's the yolk and the white. Okay, so that is the functional part of the egg, just like our neurons are the functional part of the egg. Each individual egg is surrounded by a shell, right? That shell is a protective covering, just like the endoneurium protects these individual neurons. 12 eggs are stored together in a carton, right? So a cardboard or styrofoam carton, whatever. That carton is, grouping these eggs together and providing an extra level of protection. So we can think about the carton full of eggs as a fascicle. Right? The fascicle is the whole, the dozen of eggs, the unit by which you buy things, and the carton itself is the perineurium. Finally, in order to ship lots of eggs to the grocery store, generally eggs are shipped in these big plastic, almost pallets. Okay, and so the entire nerve here is essentially like a pallet of individual dozens of eggs, right? And the plastic pallet itself is the epineurium, so providing additional structure, grouping all of these dozens of eggs or these fascicles all together for ease of transport. Okay, also note that in the neuron, so not in a pallet full of eggs, but in the neuron, there are blood vessels contained within these nerves as well. Okay. So the peripheral nervous system contains these nerves. Okay. Nerves ultimately carry information to and from the central nervous system, as we know, and so the central nervous system also needs its own protective membranes. It has a little bit of a different organization than these peripheral nerves. Okay, so again, we will be discussing the spinal cord at length very shortly, but for now, note that there are also protective membranes, just like epi, peri, and endoneurium on nerves, protecting the central nervous system. These specialized membranes are called meninges. Okay, so let's look at the meninges themselves. Because your central nervous system protects all of your stored memories, and it is the control center of the entire body, we desperately need to protect it with many different layers of protection. Of course, as we can see here, the central nervous system is entirely encased by bone. All right, that's pretty darn protective. There is a layer of fat, right? Both your brain and your spinal cord protected by fat. And then, deep to this fat and the bone, we can see three independent meninges, okay, these shock-absorbing specialized membranes. Okay, we'll go from superficial to deep, so from the top, closer and closer to the spinal cord itself. The first is the Dura Mater, so Dura for durable. This is fibrous connective tissue, so essentially dense, irregular connective tissue, allowing strength in all directions. The Arachnoid Mater, arachnoid indicating spider-like. If we look very closely here, we can see some kind of spider-like protein fibers, like the little legs of a spider here. Essentially, these proteins are keeping distance between the arachnoid and the next of the three meninges down here on the spinal cord itself. These spider-like proteins allow a space to enclose the cerebrospinal fluid. Right? So at this point, we know that CSF is bathing over the surface of the spinal cord in this kind of subarachnoid space, and it is traveling through this, the central canal within the gray matter of the spinal cord as well. So CSF on the surface and within the central nervous system. Finally, we have the pia mater, pia or pita. This is actually on the surface of the central nervous system itself. Right, so it's very elastic, lots of collagen, so giving strength to this, bundle of neurons, essentially. Again, I want to bring your attention back to this fatty tissue surrounding the entire spinal cord. There is adipose and loose areolar connective tissue providing padding to the entire spinal cord. That padding, that fatty tissue, is stored within what is called the epidural space. Of course, you have heard of epidurals before, not necessarily as a space around your spinal cord, but instead as a treatment. Right? So you've probably heard of an epidural, applied during childbirth. Essentially, what happens there is that a needle is inserted between two vertebrae, and it's going to apply medications, right? Oftentimes anesthetics, pain medications, muscle relaxants, whatever, are applied to the nervous system, the central nervous system, within this epidural space. Okay, so essentially anything from the site of application all the way down is going to be numbed, for example, or is going to receive the benefits of this medication. Okay, anything from the site of application up is going to be relatively unaffected. Okay. Another condition, another term that you might have heard of in relation to these meninges is meningitis, right? Itis for inflammation, meninge for the meninges, right? So these three meninges, Dura, Arachnoid, and pia mater, they can become inflamed with infection. Kind of the most common is bacterial meningitis. So this is actually a streptococcus bacteria, so strep throat maybe, also found in other places in the body. An infection that might start in the lungs, for example, can ultimately cross over the endothelium of our blood vessels and ultimately get into the circulation of our blood and our lymphatic tissue. Over time, if this condition, right, if this strep is not treated appropriately, if it's not removed, ultimately these bacteria can make their way up into the spinal cord, right, and can cause inflammation of the meninges on the brain as well. The brain, of course, doesn't have much room in our skull to expand, right? It's very, very close to the surface of our skull, and so any kind of inflammation will actually start to compress your brain tissue. Okay, and so this can obviously be fatal if not treated appropriately. Bacterial meningitis is certainly not the only type of meningitis. This inflammation might come from a virus, right? It might come from fungus, some kind of a parasite, the brain-eating amoeba. You might have seen in the news, this pops up every once in a while. Essentially, this is a parasite that resides in pretty warm lakes, so usually warmer than you would find in South Jersey, but Georgia, Florida, much warmer places. These amoeba can actually make their way into your central nervous system, oftentimes through your nose, right? So you're going swimming, you get water up your nose, and this causes a severe form of meningitis, which can be fatal in mere days. Okay, so just another perspective on the spinal cord. Here we can see all three of the meninges. Right, so this is looking from the posterior side. This is the bone that you feel, the little bumps that you feel on your back. See, here is the Dura Mater, most superficial. Next is the Arachnoid Mater. Deep to that is your subarachnoid space where your CSF would be circulating. And finally, the pia is directly on the surface of the spinal cord. Okay, also, what we can see here are peripheral nerves coming in/out of the spinal cord. We'll go over those in more detail in just a moment. Also, we can see the gray matter, this nice butterfly shape surrounding the central canal, right? Also with ependymal cells and CSF, and of course the white matter surrounding that. So these are myelinated axons, and this gray matter contains synapses between neurons. So again, sensory afferent information and motor information is carried to and from the central nervous system by spinal nerves. Right? We know the spinal nerves are organized with this epineurium, perineurium, and endoneurium, kind of bundling individual neurons together. Okay. Here we can see a picture of the nervous system, right? Central nervous system with the brain and the spinal cord, and we can see that all of these peripheral nerves, again, carrying afferent and efferent information to and from the central nervous system for processing. Okay. The spinal nerves themselves are going to actually get to the central nervous system from gaps in between the vertebrae. Okay. Also, there are cranial nerves which directly enter the brain via holes in the skull. Okay, but that's much later this semester. For now, we are simply talking about how peripheral nerves, how these spinal nerves get to the spinal cord. Okay, so here we can see gaps between the vertebrae, spinal nerves passing to the spinal cord through these holes, the intervertebral foramen. Okay. All right. What we can see here is a section through the spinal cord, as well as spinal nerves delivering information and carrying motor information away from the spinal cord. Sensory and motor information is delivered to and from the spinal cord in very predictable ways. Okay, so it's always the same way, and we name the structures accordingly. This here is the spinal nerve, so that's what would be passing into the spine to carry information. We'll start with to the central nervous system. Okay, so what we can see here is a sensory fiber, an afferent neuron, passes on the dorsal side of the spinal cord, so facing your back. Okay, and ultimately delivers this message, right, releasing neurotransmitters within the gray matter of the spinal cord. Okay, that information is relayed to an interneuron, right? Sometimes an interneuron might carry the information up to the brain, right? So maybe traveling back here in the white matter and going straight up. Or, in the case of reflexes, an interneuron can immediately transmit this message to a motor neuron. Here's a motor neuron, motor fiber, exiting the spinal cord on the ventral side, okay, and then joining together with the sensory fibers to exit the spine and go out to the rest of the body. Okay, so let's add a couple names here. On the dorsal side of the spinal cord, we have what is called a dorsal root. Okay, this dorsal root contains only sensory neurons, right? No motor, only sensory. On the ventral side, we can see only motor fibers. No matter what, only motor information. It is not until the spinal nerve itself where we get both sensory and motor neurons together. Okay. When you are looking at a model, for example, or even an image of the spinal cord, you can tell the difference between the dorsal root and the ventral root by a little bump. Okay, there's going to be a little bump called a dorsal root ganglion, which is present only on the dorsal side. And this dorsal root ganglion is a group of neuron cell bodies, specifically a group of sensory neuron cell bodies. Remember that the sensory neurons are mostly unipolar. Okay, so here we can see one little appendage off of the cell body of the neuron. They're all stored pretty much right outside of the spinal cord, making up this big grouping right here. A ganglion is cell bodies grouped outside the spinal cord or outside the central nervous system. So here is a dorsal root ganglion. This little bump is only present on the dorsal side. Right, knowing these structures is going to be important for both the lecture and the lab. One way to remember which side, right, the dorsal or the ventral, actually has the sensory and motor information is Dave. Okay, so Dave, D for dorsal, right, the dorsal root, carries only afferent information, whereas the ventral root, right, the ventral side, only contains efferent information. Dave. Okay, this here is a little bit of a nicer view of the spinal cord. Again, what we can see is the dorsal root ganglion, right? This little bump only present in the dorsal root for afferent information, and here would therefore be the ventral root. Okay, the gray matter inside the spinal cord, right, is where all of these synapses are occurring. On the dorsal side, this is where all of the sensory information is first relayed to those interneurons. There are actually different places, different regions of this gray matter where synapses occur for the visceral sensory information versus the somatic sensory information. Of course, when you look at, a dissected specimen or a model or something, it looks just like a bunch of gray, but in fact there is exquisite organization within this, otherwise just gray stuff. Okay. On the ventral side, this is where all the synapses occur for the somatic motor decisions, right? So what our skeletal muscles are doing, and your visceral skeletal visceral decisions. Okay. So again, lots and lots of organization within the gray matter. Synapses between neurons regarding certain places within the body are very much organized within this. Okay. We can name different structures within the gray matter. Right? So up here where all of these sensory relaying, or the sensory information is being relayed, this is called the posterior or the dorsal horn. Right? So just this shape of gray matter. Okay. On the other hand, we had the anterior horn, right? This front kind of part of the little butterfly shape of gray matter, and finally sticking out to the sides, only in certain places of the spinal cord, are the lateral horns. Right? I say only in certain places of the spinal cord because if there isn't a lot of visceral information coming in, right, that is, if you're not carrying information about the heart or the lungs or the stomach or whatever, there is not a lot of gray matter in this area. Just like the gray matter is organized, the white matter is also organized. Again, so when you look at this with the naked eye, you do not see all of these different well-defined regions, but in fact, we know that axons carrying information, about certain types of stimuli are carried back here on the dorsal side, some are carried all the way on the lateral side, some on the anterior side. That we are going to focus on all the way at the end of this class, but I do want you to be aware for now that there is organization within the gray matter and the white matter regarding specific types of information and decisions. The images that we've been looking at have just been kind of a generic section through the spinal cord. However, as you might imagine, not every region of the spinal cord is going to look exactly the same. In fact, there is considerable variation in spinal cord structure between whether you look all the way at the top of the spinal cord versus all the way at the inferior side of the spinal cord, right? So a lot of different variability. And first of all, the shape of the gray matter kind of butterfly and the white matter surrounding it. All the way at the top in what's called the cervical region of the spinal cord, the shape does not generally include lateral horns. Okay, remember that the lateral horns, right, so this lateral projection of the gray matter, this doesn't exist because there is not visceral information coming into the spinal cord. That is, there aren't internal organs in your neck. Right? On the other hand, lower in the spine, or lower in the spinal cord, here's where your heart and your lungs would be, and here is where your digestive, reproductive, urinary organs would be way down here, and so there's a lot more visceral information coming in what's called the thoracic and the lumbar regions of your spinal cord. So if we look at for example, thoracic, right, your back region of the spinal cord, we see very clearly defined lateral horns. Okay, we also see pretty clear lateral horns within the lumbar as well. Secondly, your spinal cord, right, and all of these different structures within it, right, the different columns of white matter, the posterior versus ventral horns, all of these different structures stop partway down your spine, right? So these do not go all the way to your tailbone or your coccyx. Instead, they stop at about L1-ish. So pretty much right under your rib cage. Right? The rest of what is contained within your spine, including all the way down to your sacrum and coccyx, is what is called the cauda equina. Cauda equina means literally the horse's tail, right? Equine for horse, cauda, caudal for tail. So this structure, again, is not the same as your spinal cord. It is merely a bundle of spinal nerves. Okay, so let's kind of take a step back here. What is going on? For about the first 4 years of your life, your spine and your spinal cord grow at about the same rate, right? So as you go from the size of an infant up to a toddler, right, you're 4 years old, your spinal cord and spine are growing together. However, your spinal cord stops growing at about 4 years old. But of course, your spine continues to grow until you're a teenager and beyond. And so, in order to, so your spine continues growing, right? And what continues to grow inside your spine are the spinal nerves, right? So what we can see here is that individual spinal nerves are still exiting out of that spinal cord, but in order to actually exit the spine between the appropriate pair of vertebrae, the spinal nerves have to grow and grow and grow. And so at the end of your spinal cord is essentially a bundle of spinal nerves. Right? So here, a bunch of spinal nerves, each exiting the spine in their appropriate location, but definitely not being the spinal cord. Right? So this bundle is called the cauda equine. Finally, within the spinal cord, there are 31 segments. Right? They're not actually divided by lines or anything. These are kind of hypothetical segments, but there are 31 segments nevertheless. Each of these 31 segments is associated with the 31 spinal nerves. And as we saw in the previous slide, the spinal nerves exit between a pair of vertebrae or within holes within the sacrum or coccyx. And so each of those spinal nerves actually is going to innervate, right? Detect stimuli, send motor information to a very specific region or a very specific grouping of structures. Each of the 31 spinal nerves is named according to where it exits the spine. Okay, we'll talk about that in a moment, but we can see for now that there are, C2, C3, T7, T8, S1, right? So all of these are exiting the spine in a very specific spot. Okay, these spinal nerves are also innervating a specific grouping of structures. So this might be skeletal muscles, but also its own region of skin. And so that is what is mapped out on these diagrams here. These are called dermatomes. The nerve T3 innervates this little strip across the back, right, and this little strip across the ventral side of the body. Right? So this is important because infections of the spinal nerves or infections of the dorsal roots or part of the spinal cord, if there is some kind of damage, right, if you're in a car accident or something and damage to your spinal cord, you can figure out exactly where that damage or where that infection is located. If you lose feeling or have pain or other signs of infection within a certain strip of your skin. One example of this is shingles. Shingles is a type of virus that infects specifically the dorsal roots. Oftentimes this is a result of chickenpox. Having chickenpox when you're younger, it can remain dormant within your dorsal roots, and later in life it can make an appearance again. Oftentimes a lot more painful, a lot more severe than the initial chickenpox infection. Shingles often presents as red patches on the abdomen, right, or on other parts of the body, but abdomen is a really common location. And oftentimes these little red patches are in lines, right, so only within particular dermatomes. And so what we can learn from that is exactly where the infection is located. Right, so if the red patches are only over here, right, you might understand from that that this virus is located in the dorsal roots of T8, T9, and T10 only. Okay. What we can see in this image is the spinal cord. Right, we can see the dorsal and ventral roots. Right, we can see the dorsal root ganglion only present on the dorsal side. Of course, where the dorsal and ventral roots come together, we have the spinal nerve. Okay, so we know the spinal nerves enter and exit at very specific spots of the spine. Once those spinal nerves exit the spine, okay, they often split, they bifurcate nearly immediately, sending some of the axons within them around to the back of the body and, some of them around to the front. Okay, so we have names for that, of course. So here, the spinal nerve, we can see is splitting. Again, some axons come back here, and some are going to go all the way around to the front of the body. This particular region is within the thorax, so each of these little bundles of neurons is traveling with a rib, right? So we can see that they are parallel to each other. Okay. The branch of the spinal nerve that is traveling around to the posterior side of the body, ultimately innervating the muscles of the back, innervating the skin of the back. This is called a dorsal ramus, so dorsal for back, ramus for this branch of the spinal nerve. Okay. Most of the spinal nerve, however, is going to travel around to the anterior side of the body, and that is called the ventral ramus. Okay, ventral ramus down here. Furthermore, the spinal nerve, depending on where you're looking in the spinal cord, can also divide or send axons, branches of the spinal nerve, along something called communicating or sympathetic rami. Rami for plural, ramus for singular. So what we can see here is extra little branches here that are ultimately sending axons to travel parallel to the spinal cord as part of your sympathetic nervous system. Okay, that's a topic for later this semester and surely for next semester. But for now, we have lots of branching of the spinal nerve. The dorsal ramus goes to the back, the ventral ramus goes to the front, and the communicating rami are going to communicate with this parallel sympathetic chain of ganglia. Okay, just another way of showing you this. Again, the spinal cord with the posterior and anterior horns of the gray matter, dorsal root, ventral root, come together to form the spinal nerve. Spinal nerve splits, it branches into the dorsal ramus going to the back, the ventral ramus going to the front, and the communicating rami literally communicating with part of the sympathetic or part of the autonomic nervous system. There is a model in the lab, and I just want to point out a couple of things on this model now so that you kind of know what you're looking at. Okay. Spinal cord within the spine. Here we can see some of the ribs, right? So this must be in the thoracic region. We can see gray matter a little bit up here surrounded by white matter. We can see dorsal and ventral roots coming in and out of the spinal cord. A branch goes around to the back, that is the dorsal ramus. The ventral rami are going out to the side, essentially traveling with the ribs. Okay. And finally, the communicating rami are literally communicating with this sympathetic chain of ganglia. Here's one, and here's the second. Okay, so definitely make sure you spend some time with this model. Some more branching. Okay, so lots of different organizational patterns of the nervous system in the thoracic region. Right, so in the region of your spine that contains the rib cage. Each ventral ramus is going to travel with each individual rib. Right, so one for one. Everywhere else along the spine, there are what are called plexuses. Right, so multiple plexi. These are essentially a series of ventral rami from multiple spinal nerves that instead of traveling in a straight line along a rib like in the thoracic region, instead they join together again, right? Multiple spinal nerves split apart, join together and split apart, and essentially create a mesh, an interwoven network of nerves instead of individually traveling nerves. Okay, so we can see that here. Here's the cervical plexus, right? It's a branched network. Here's the brachial plexus, again, lots of branching. And again, we can see lumbar and sacral. Everywhere throughout the spine and spinal cord, we get a plexus except for where the nerves are traveling with the ribs. Essentially, this is, or these structures are not there to make our lives harder. Right? They are actually there to more or less act as a fail-safe. Right? So if you break your arm and you sever one of these nerves, right, it's not the end of the world because there is still information traveling, from spinal nerve number, T1 in other parts of this plexus. Okay, so definitely helps us out in that regard. Okay. There are several, 4 main plexuses that you guys should be familiar with. The cervical plexus, way up here in the cervical region, the brachial plexus essentially innervating down the brachial region, right, the upper arm. The lumbar, again, the lower back, and the sacral around the sacral region, around the sacrum bone. Okay, so hopefully these terms aren't new to you, right? These are actually the same regional terms that you learned already. As I stated on the very first day of class that we had together, in my section of AMP1, we study the body in regions as opposed to in pure systems. And so, in our study of the spinal cord, let us also study the bones that are surrounding the spinal cord. Right? So, the axial skeleton, right, is comprised of bones that are essentially protecting the central nervous system, right, and establishing the axis of the body. Okay, what we have is the skull, and we will focus on that extensively at the end of this semester. The spine, right, including 24 vertebrae, the sacrum and the coccyx, and the rib cage, right, the rib cage and the sternum together, all generating or all making up the axial skeleton. Okay, this part of the skeleton is responsible for supporting and protecting the entire central nervous system, protecting the internal organs, right? So again, the axis of the body. Finally, it is the attachment site for many muscles. We can stand up straight because muscles are pulling on bones within our axial skeleton. Okay, this here is our spine. Again, we have 24 vertebrae. Okay, they are named according to what region they are found in. So we have 7 cervical vertebrae. They are named C1 through C7, right, according to their position, starting with the most superior bone. Next, we have 12 thoracic vertebrae. So T1 all the way down through T12. Finally, we have the lumbar vertebrae, so L1 through L5. At the most inferior part of the spine, we have the sacrum, which is actually 4 or 5 fused vertebrae, and they were fused during our own development as well as evolutionarily. Okay. And the coccyx is your tailbone, which is also a series of fused vertebrae fused during our own development. Each vertebra articulates or forms a joint with each other in several different places. First of all, the bodies of the vertebrae, so this big weight-bearing portion of the vertebra, the one that is pointing anteriorly, so this is pointing towards towards your stomach, towards your viscera. The bodies are separated by what are called intervertebral discs. These are shock absorbers, so whenever you're doing jumping jacks, your vertebrae aren't compressing and grinding against one another. Okay, they are made of something called fibrocartilage, which we'll talk more about shortly. Also, we can see that the vertebrae, even though they articulate here at the bodies and they're also articulating back here, and we'll look at that soon, there's always a hole in between them, and this is called the intervertebral foramen. Between vertebra, right? So between the vertebra. And this here is a word that we will see again and again, lots within our study of the skeleton. Foramen or foramen means hole, right? So this is a hole literally in between the vertebrae. As we know, this is where the spinal nerves are going to exit the spinal cord and travel to the periphery of the body. Again, fibrocartilage is a type of cartilage. Cartilage happens to be a type of connective tissue. Okay, this is some histology of fibrocartilage. Right, we can see this kind of milky texture right here. That is essentially a lot of fibers, a lot of collagen, a lot of proteoglycans, which are essentially trapping water and creating this really squishy textured material. Okay, fibrocartilage in particular has a lot more collagenous fibers, lots more collagen to make this particularly tough and rigid. Okay. Also, within this type of connective tissue, we need to have some specialized cells, right? Just like we saw with any other type of connective tissue. These specialized cells are called chondro. Chondro is a root for cartilage, cyte for cell. Okay, so these are literally the cartilage cells, and they live in what are called lacunae. These little holes here essentially trap the chondrocytes, and they reside within these little cavities, within these little holes, for the entire duration of their life. Okay. One of the other features of cartilage in general, not just fibrocartilage, is that it is relatively avascular. Okay, so what this means is that if you damage your cartilage, it is possible for it to repair itself, but it takes a really long time, right, just because it doesn't get many nutrients. Okay. The vertebrae all share particular characteristics with one another. I'm going to walk you through them here. This is really something that we focus on in the lab, but I want you to have a pretty good understanding before you actually see these vertebrae in the lab. This happens to be a cervical vertebra. Here we're looking from the superior side, here we're looking from the lateral side. The body is the weight-bearing portion of the bone. There is a hole just posterior to that weight-bearing portion, and that is called a vertebral foramen. Okay, so a hole through the vertebra. This is where the spinal cord lives. Okay. There are also a series of processes. Right? A process is an extension of the bone, so it's sticking out away from the main portion of the bone. Pointing directly posteriorly is the spinous process. Right? So if you run your finger down your spine, through your skin, you can feel these bumps. Those bumps are the spinous processes of your vertebrae. Okay. We also have transverse processes. Right? These stick out in the transverse direction, so essentially making a nice horizontal plane of bone. And finally, there are articular processes. Right? So one portion of the bone sticks straight up, another portion of the bone sticks straight down. We can see this a little bit better from this perspective. Right? Superior process is going to stick up, inferior process is going to stick straight down. So essentially what these articular processes are doing is providing a site for articulation, right? So forming a joint with superior and inferior vertebra on either side of this one. Okay. Also here we can see facet. This light blue circle here, the light blue is indicating that this is cartilage, and essentially this cartilage indicates that this is forming a joint. So the facet itself is where the two vertebrae join together. Okay. Final structures here. Around the vertebral foramen, we can see that there's essentially a circle of bone made up by the body for the large part and a little bit the spinous process, but also we have what are called pedicles, right? So between the transverse processes and the body, and we have a lamina, so lamina here and here, ultimately making a ring of bone around the vertebral foramen. Okay. Let's see how these articular facets actually come together. Okay, what we can see here is a spine that is both bending forward, flexing, and bending backwards, extending. Okay, we can see the intervertebral discs essentially are these big cushions, and they allow a reasonable amount of flexibility between the bodies as well as acting as shock absorbers. But also we see the inferior articular process and the superior articular process are joining together between these two vertebrae. So that is where the joint actually exists. Right? When we flex and extend our spine, these joints essentially slide over one another, right, facilitating this movement. Okay. From another perspective, this is the posterior side of the spine. Again, these are the spinous processes. We can see the transverse processes sticking out to the side. Each vertebra has a superior and an inferior articular process, and here we can see the two superior and inferior processes articulating together to form this extra joint. So 3 ways that each pair of vertebrae join together. Okay. When we join multiple vertebrae together, their vertebral foramina for plural foramen, they join together and form a canal. Again, this vertebral canal is where the spinal cord is. I also want to point out the intervertebral discs. They are mostly fibrocartilage, but all the way on the inside. There is an inner fluid-filled compartment called the nucleus pulposus. We'll see that come up again. Okay, there are a few unique characteristics among the cervical vertebrae. So on a lab practical, I could put a vertebra on the table and say, what region is this from? And you would have to tell me cervical, thoracic, or lumbar. Okay, so how exactly do we tell the difference between cervical and the other two? First of all, the cervical vertebrae only, among all of them, are the only ones to have what is called a bifid spinous process. The spinous process actually forms a little bit of a Y, and we'll look at why that is in the lab. But that forms a Y, whereas this does not happen in any other region, that is. Also, a transverse foramen or transverse foramen. There are holes in the transverse processes only of the cervical vertebrae. The reason for this is that there are blood vessels that travel encased within these bony holes. Of course, in order to get blood up to your brain, which is very hungry, needs lots and lots of blood, the blood has to travel from protected within your thoracic cavity all the way up your neck, which is very much exposed to finally get to your skull. And so we protect the blood, ultimately nourishing our brain, by sending some of it up through our spine, which, as we know, is very protected. Right. Finally, the cervical spine, C1 through C7, contains 3 vertebrae with special names. Okay, so vertebra C1, C2, and C7 each have a unique name: atlas, axis, and the vertebra prominence. Down here in the corner, we can see atlas and axis. Atlas, named after the Greek god holding the world on his shoulders, is C1. So essentially, this vertebra holds our world, our head, on our shoulders. Okay, axis, we can see here, actually forms a pivot point, and atlas can actually rotate around this, and I will show you this in the lab. The vertebra prominence, we can see down here, this is C7. This is the most prominent vertebra of your spine, and essentially we can feel this through the back of through the skin on the back of our neck, and it's clinically significant because this essentially signals the end of the cervical spine and the beginning of the thoracic spine. Okay. One more note before we move to the next slide. Up here are some instructions on how to get to some resources on mastering A&P. In your study area under lab resources, we have Practice Anatomy Lab, PAL, and there are a series of images that you can play with to get a good feel for what the structures on these vertebrae are. Just a little bit more about atlas and axis. Atlas, because it is holding up your skull, so articulating with your skull as opposed to with 2 vertebrae, it is structured a little bit differently. Namely, there's no body. Right? These articular facets are huge, right? They're articulating with your skull. Also, the vertebral foramen is very large, right? Your entire spinal cord has to pass through this, right after it exits your skull. Axis essentially has taken the body of atlas and fused it with its own body. And so there is a process called the DENS, or sometimes called the odontoid process, that acts as this pivot point. Okay, and finally, the vertebra prominence has a very prominent spinous process. Okay. Now, exiting the spinal cord in the cervical region are not 7 cervical vertebrae or cervical spinal nerves, but 8 cervical spinal nerves. Right? So there are 7 vertebrae and 8 nerves. We name spinal nerves C1 through C7 according to which vertebra is inferior to it. That is, spinal nerve C1 exits on top of vertebra C1. Spinal nerve C2 exits on top of C2. C3, C4, C5, C6, C7. And so here's the vertebra prominence C7. Now the next vertebra in line is T1, the thoracic vertebra number 1. However, in between C7 and T1, the vertebrae exits an 8th cervical spinal nerve. This is called C8. Okay. Therefore, from vertebra T1 all the way down to the end of the spine, each spinal nerve is named according to what vertebra it is under. All right, so T1, the nerve, exits inferiorly to vertebra T1. Okay, so again, cervical spinal nerves, C1 through C7, exit superiorly to their vertebra. C8 exits inferiorly, and all of the rest of the spinal nerves all the way on down exit inferiorly to their nerve, or to their vertebra. Okay. Thoracic vertebrae, what makes them unique? Well, first of all, the thoracic vertebrae, the 12 of them, each articulate with a pair of ribs. Right? There's 12 pairs of ribs, there are 12 thoracic vertebrae. Okay, so what that means is that there are a couple extra facets or articulation points on the thoracic vertebrae as opposed to others. These are collectively the costal facets. Right? So costal means rib, and so we have one costal facet on the transverse process and one costal facet on the body of the vertebra. As we can see down here, the rib articulates with the body and with the transverse process. Okay, another feature that's pretty distinctive about the thoracic vertebrae is that their spinous process points roughly inferiorly. Okay, we'll take a look at this in the lab, but for the most part, we need to have these inferiorly facing spinous processes so that we're not essentially a stegosaurus. Right? We don't want these processes sticking through the skin of our back. We want them to kind of fold over one another to create a smooth spine. Finally, the lumbar vertebrae. When you see these in the lab, you will see that these things are just huge in comparison to all the other vertebrae. So the size alone should be able to help you distinguish between lumbar versus the other two regions. But also, the spinous process, the lumbar is fairly flat and hatchet-shaped. Right? So it points posteriorly instead of inferiorly. So that's pretty distinctive as well. The body is very large as well, and the angle of these articular processes / articular facets. The lumbar spine has very limited mobility, particularly when it comes to rotating, right? So kind of swiveling back and forth. That is because these articular processes face medially on the top and laterally on the bottom. And so again, as we'll see in lab, the lumbar vertebrae kind of fold into one another. Instead of being able to slide left and right, up and down, they can only slide up and down. Okay, and again, we will play with that a bit in the lab. Okay, all the way at the bottom, so the caudal end of the spine is the sacrum. As I said before, the sacrum or sacrum is a series of fused vertebrae. Again, so you can actually see some of these features as a tie-over for when these were individual vertebrae. Particularly, we can see on the posterior side of the sacrum this ridge here. All right, these used to be the spinous processes. Now we call them the median sacral crest. Okay, we can see these holes, right? The intervertebral foramina. Now we call them sacral foramina. It just holes within the sacrum. And finally, that vertebral canal becomes the sacral canal. So the end of the cauda equina actually passes into the sacrum, still allowing nerves to exit through these holes. Okay, finally, the coccyx or the tailbone, again, about 4 fused vertebrae. Final portion of the axial skeleton that we're going over now, that is the rib cage. Okay. The ribs on the anterior side articulate via their own cartilage or a combined cartilage or no cartilage at all with the sternum. The sternum contains 3 different parts. This is your quote-unquote breastbone. The manubrium is this part up here. This is the body. And finally, the xiphoid process is cartilage, right? But ultimately it will become calcified over time. And so one of the ways that archaeologists can age a body is by looking at the xiphoid process. Okay, so how much is still cartilage, how much is bone? Okay. Next, the ribs. Right? We have 12 pairs of ribs. The first 7 pairs of ribs articulate, of course, directly with their vertebrae in the back, right, with those costal facets. And on the anterior side, they're going to articulate with the sternum via their own independent costal cartilage. Okay. This particular joint has a relatively long cartilage so that whenever you breathe, your chest kind of changes shape a little bit. There's a lot of flexibility and a lot of room for expansion of your thoracic cavity with these relatively long cartilages. Okay, so 1, 2, 3, 4, 5, 6, and 7 all have their own costal cartilage to attach to the sternum. The remaining 5 pairs of ribs are called false ribs, so ribs 8 through 12. They either have an indirect connection with the sternum, so here we can see 8, 9, and 10 all have fused costal cartilages. These are false ribs. Okay. The last 2 pairs of false ribs, so this one here, 11 and 12, 11 and 12 don't actually connect to the sternum at all. Okay, these ones are going to just kind of be floating on the posterior side of your thorax. They are more or less protecting your kidneys in the back. Okay, so again, true ribs have their own costal cartilage. False ribs share a costal cartilage or have none whatsoever. Finally, this is just a summary of the entire vertebral column. Note that there are, of course, 24 vertebrae, right? 7, 12, and 5. At the inferior aspect, we have the sacrum and the coccyx. Okay, each vertebra is joined together by the intervertebral discs as well as these articular facets. Okay, finally, the intervertebral foramina are where the spinal nerves are going to exit the spinal cord and exit the spine. Remember that these spinal nerves are named according to their adjacent vertebra. So for example, C1 through C7 exit superiorly to their vertebra. C8 exits between C7 and T1, and T1 all the way down to the coccygeal nerves. All of these exit inferiorly to their vertebra. Okay. One more note to make on this slide. Curves. Note that there are convex and concave curves within the spine. Note that the thoracic curve and the pelvic curve, which is the sacrum and the coccyx, are both curved towards the anterior, and the cervical curve and the lumbar curve also traveling in the same direction but exactly opposite from the thoracic and the pelvic. Okay. We can call the thoracic and pelvic curves the primary curvature. Essentially, this is called primary curvature because this is established in the womb. Okay, so when you are quote unquote in the fetal position, your spine is curved in this direction, right? A big C shape is how your spine initially forms. Okay, and you remain with this curvature for the first few months of your life. Over time, however, as long as everything's working correctly, you start to crawl, you start to lift your head up. And so the cervical curvature develops, right? The secondary curvature as you lift your head up, right? And this kind of balances out this curve of the thorax. And of course, as you start to crawl and ultimately stand, the lumbar curve, right? Again, in the opposite direction from the pelvic and thoracic, develops secondarily, again, as you start to walk on 2 feet. I give you guys these links just because at this point perhaps you can appreciate this a little bit more. This is a news article from a couple years ago. An Australian toddler was in a car accident with his mother. He was internally decapitated, that is, his C1 and C2 separated. His spinal cord still remained intact, but the physicians were actually able to reattach this toddler's head. At this point, the kid is perfectly healthy. So I just want to point out that, if you're going into nursing or PT or anything, you might be dealing with patients and helping them to recover who are decapitated. We are at that point in our scientific development that we can reattach people's heads. Right? So kind of cool stuff there. On that same note, speaking of reattaching heads, just a couple notes about this human head transplant topic. Dr. Sergio Canavero, a few years ago, made the claim that he was going to do a head transplant, and here is his volunteer head. This particular volunteer has a degenerative muscular disorder, and so in order to function, he is completely dependent upon other individuals, but his mind is completely fine, and so he has volunteered to donate his head to be transplanted onto another individual. And so there's a ton of references here and talks, when this first started making the news, about this procedure. So since Canavero first came out and said that he was going to do this head transplant, he's done some work, albeit he has not been very transparent about it and has not followed the normal ethical guidelines required of medical professionals and scientists. But regardless, he has supposedly been able to do a head transplant on a monkey in 2016. Okay, we do not know the fate of this monkey at this point. To the world's knowledge, he hasn't done any further experiments exactly like this. So he did it once. Supposedly it was okay, but that's where that ends. And so here is some of the limited information that has been released. In the same year, he supposedly glued back together a spinal cord that he severed of a dog. Supposedly, the dog was perfectly fine within just a couple weeks. Okay, again, we don't really have a lot of information about it. This is the minimal amount that's actually been released. Next, he was able to surgically implant a second head onto a rat. Okay, and supposedly both heads were functional. Okay, so in theory, getting closer and closer to doing a human head transplant, perhaps. 2017, right, October 2017, he claimed to have, quote unquote, successfully completed a transplant on a corpse, a human corpse, that is. And so there are a number of issues with this, aside from the ethical implications of this. First of all, transplants, right, they always begin working with cadavers, right? But in order to actually move to actual transplant, a successful transplant, you need to work with living individuals. Like, a successful transplant is not successful if it is on to corpses, right? By definition, this is not a successful transplant. Also, it takes dozens, hundreds of trials in order to be considered successful. And Popular Science has a very snarky article about the science behind this man's techniques. Definitely kind of an entertaining read. But anyway, by normal standards, normal transplant standards, this was not a successful trial. He did this once. He sewed a head onto a body. No way of knowing if it actually would work in a living individual. Okay. Final note to make on this. Last year, supposedly the volunteer got married and changed his mind about actually being the head donor for this. The surgery is incredibly risky. Even if he did survive the surgery, he might be in excruciating pain. He might not be able to work his body anyway, right? He might be paralyzed for the rest of his life, regardless of having a new body. And so, Now there's no head donor, and heaven knows where the body donor would have come from for this surgery in the first place. But according to very limited sources, again, it's really difficult to follow what is going on with this. They're being very elusive. Supposedly, Canavero has moved to China, where there are fewer regulations on these types of things, and he and a Chinese surgeon are planning on continuing with this effort. Okay, so to be continued. I bring this up not because, it's wild and crazy and there are so many ethical considerations that had to be taken into this, but as future medical professionals, again, you could be working with not only people who have their heads reattached from car accidents, but people who have a head transplant, right? This is revolutionizing medicine as we know it. So I just wanted to throw that out there. Take a look if you're interested. And that is the end. Okay, and so I just wanted to throw a couple questions at you guys to help you start thinking about this information. So I'm going to run through these with you super fast. I definitely recommend trying these on your own. All right, so number one, what doesn't carry sensory nerve fibers? Well, most nerves in your body are mixed, which means that they have both sensory and motor neurons, so taking information to and from the central nervous system, with few exceptions, one of those exceptions being the ventral roots. Remember that these are taking efferent or motor information from the spinal cord, not sensory information at all. There are a couple different types of neurons. These neurons are generally multipolar in the central nervous system. They're generally unipolar if we're talking about sensory, peripheral neurons, and there are very specialized, very rare neurons that are called bipolar neurons, and they're only in the special senses. Which of these neuroglia, so specialized helper cells within nervous tissue in particular, line cavities within the brain and the spinal cord? That would be the ependymal cells. What is present within the dorsal root ganglion, giving it a swollen appearance? Well, always ganglion implies cell bodies. We have ganglia all throughout our body, and you will hear a lot more about them later this term. Fascicles are bundles of axons. Which layer of tissue partitions the nerve into fascicles? Well, there's endo, peri, epi being all the way on the top, endo being all the way inside, and so in the middle we have this perineurium. The perineurium goes around the bundles of axons. Which spinal nerve exits inferiorly to T7? Everything inferior to C7 is going to be named according to its more superior vertebra, so that would be spinal nerve T7. Okay, and so I want to end today with having you guys watch a short video from someone else, different perspective, always helpful, on reflexes. And so as you're watching this video, you're going to learn about reflex arcs. You should absolutely take extra notes on the slide. I've given this to you just to help guide you through and think about the things that are most important. So think about all of the different components of reflex arcs, what reflex arcs actually are. Now, what the video doesn't mention is that reflexes are, of course, tested by physicians. And so there's not just like one reflex test that is going to indicate health in your entire nervous system, right? So we have a knee-jerk reflex, which of course is what you see on the slide right here. Also, it's what the video after this is going to talk to you guys about. And so yes, there's a knee-jerk reflex, but there are also so many other reflexes that can be tested. So we'll talk about that first. And so each individual reflex tests different nerves, right? So the knee-jerk reflex is going to test a different reflex than your calcanea reflex when you take the little hammer and you hit back here, which is different than the Babinski reflex, and so on and so forth. And so if a reflex test comes out swell, right, so your reflexes are fast, they are strong, they are not delayed, that indicates that all of the different things the things that he's going to list in here are actually in good working order. But then you have to do more reflex tests to see if other reflexes and other nerves and those other structures are also intact, right? And so, if you've ever been an EMT or ridden in an ambulance or whatever, they always check your pupils to make sure that your pupils will dilate when a light shines in them. Essentially, this is one of the many reflexes that's testing actually a couple cranial nerves, right? So if there's a delay or it doesn't work at all, there could have been an injury or there's still some sort of issue with those particular nerves and other structures. Okay, so that is what we use reflexes for clinically, right? We can test reflexes, but certainly reflexes did not evolve and don't exist in our body today so that we can hit each other with hammers, right? That's just not why they exist. And so the function of reflexes is a different question than, why do we test reflexes, right? So the function of reflexes is more or less to protect ourselves, right? First and foremost, so the knee-jerk reflex is actually an evolutionary response to essentially being a little clumsy, right? So if you're walking along and you trip over something, essentially that is going to stretch this muscle right here, your quadriceps muscle, and of course in the process stretch this patellar ligament here. And so, of course, if your toes catch on a rock, we want to immediately kick that leg out to catch ourselves before we fall flat on our faces, right? And so this reflex evolved to essentially not send the message to your brain that, hey, we just tripped over something. My quads just stretched and I wasn't expecting that. Hey brain, what do you think we should do about that? And the brain says, okay, I guess we'll kick our leg out. And then of course, this message has to go down your spinal cord, out your ventral roots, all the way out these motor neurons, all the way to your quadriceps and hamstring muscles. And so that process, while it is relatively fast and we don't necessarily experience it, it's not fast enough. And so these reflexes, including the patellar reflex, have evolved to essentially not stop and ask the brain for permission. If we trip, we kick our foot out and protect ourselves. Right? And of course, the brain will learn about this only after it happens and after we are safer. Right? If you put your hand on the hot stove, you immediately lift your hand up. Right? You don't want to stop and ask the brain, hey, it's hot, what should I do? No, if your hand is super hot, you are going to remove your hand immediately from that source of heat. And so again, the function of reflexes in the human body is not to be tested. We use them to learn a lot, but that is not their function. The other function of reflexes is to respond quickly to changing conditions, right? So for example, if somebody gives you a bag, right, in your hand, you might be subconsciously expecting the bag to be really light, but in fact there are 3 textbooks and a laptop in the bag, and all of a sudden, your muscles aren't prepared for that excess weight, and so you have a fraction of a second where, you are adjusting, right? Your hand starts to go down with that extra weight, but then you immediately respond by contracting those muscles and lifting the bag up accordingly. And so that is reflexive as well, right? If there's an additional stretch on your biceps brachii muscle or your bicep muscle, it will immediately contract and therefore respond to these changing conditions. And of course, we can teach ourselves a lot of different reflexes, right? So if you play an instrument or you are an athlete, right, you automatically see the ball out of the corner of your eye and respond accordingly without even thinking about it, without even stopping and asking your brain for permission. You immediately reflexively respond. And so no thought down here essentially is showing you that you're not asking your brain. Right? So those are some key components of reflexes that you should be aware of. All right, so at this point, watch the video, right, and then watch the lab videos. Thanks so much, everybody, and have a wonderful day.